Well, good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Maximizing Value in Early Phase Oncology Trials Innovative Endpoints for Early Insights. My name is Samya Shashikumar, and I'll be your X Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel and that can be found on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. I would also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the comments tab. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming to those who registered on xtalks.com. At this point, I would like to thank Clario, who developed the content for this presentation. Clario is a leading provider of endpoint data solutions to the clinical trials industry, generating high-quality clinical evidence for life sciences companies. They offer comprehensive evidence generation solutions that combine medical imaging, ECOA, precision motion, cardiac, and respiratory endpoints. And now I would like to introduce our speakers for today's event. Dr. Todd Rudo provides medical and scientific leadership across Clario. He brings nearly 20 years of experience in clinical cardiology and pharmaceutical research, with much of his career focused on drug safety. His team supports clients on scientific and regulatory strategy and helps ensure Clario's solutions generate reliable, high-quality clinical trial endpoint data. Dr. Anu Bansal leads medical imaging activities for oncology drug and device trials at Clario, spanning phase 1 through phase 4. He supports imaging strategy from protocol design through regulatory submission and post-approval monitoring. With more than 20 years of experience in clinical research and patient care, he works closely with cross-functional teams and clients across medical, scientific, and regulatory affairs. Dr. Kelly Dumay is a scientist with more than 15 years of experience in behavioral and life sciences research. She specializes in electronic clinical outcome assessments, or ECOA, and in developing and validating patient-reported outcomes to support labeling claims. She also leads efforts in site rater and participant training to improve the accuracy and consistency of clinical outcome data. And Dr. Robert Kleiman is a clinical cardiologist and cardiac electrophysiologist with experience in both basic cellular and clinical electrophysiology research. At Clario, he provides cardiac safety consulting to support drug development, including trial design, cardiac safety assessments, data analysis, and regulatory strategy. And with that, I'll hand things over to our first speaker and today's moderator, Dr. Todd Rudo. You may begin when ready.
Great. Well, thank you, Samia, for the very kind introductions. Thank you to all of you, our quite sizable audience that has joined us today as well. I'm excited about this one. There has been so much evolution in the past decade with endpoint solutions and strategies in oncology development. So we have certainly quite a bit to cover. You can go to the next slide. We're going to take you through this agenda today after a brief, one more slide. After a brief introduction on what's unique in early phase oncology development, Dr. Banzel will cover the evolving role for centralized medical imaging and some innovative methodologies such as tumor growth kinetics, things that can enable early insights, specifically in these small early phase studies. Dr. Dumay will speak on the value of patient-reported outcomes, that we can get out of early phase designs, specifically looking at safety, looking at quality of life. And then Dr. Kleiman will talk through some of the unique challenges to assessing cardiac safety in oncology and how to appropriately tailor a risk management strategy to your program. So next slide, please. First, just to get an understanding of this audience's perspective, we asked you all by a survey at registration, what do you see as the greatest challenges in early phase oncology development? Is it a concern with measuring impact to quality of life, to patient burden, Is it how can we best gain early insights to treatment efficacy? Or lastly, how to best measure safety risks? You see the results here on the screen. Generally, this audience was most concerned with accurately assessing efficacy and safety in early phase. I think that makes sense. And then a minority considered their greatest challenges as addressing patient burden or their impact, understanding impact to quality of life. These are all areas that we're going to cover in the next hour, but this context for this audience's mindset is certainly helpful for us as we go through today. So thank you for responding there. Please do answer, I should say, your questions in the chat as we go. We're going to do our best to get to everything at the end and leave at least 10 minutes for that. Can go to the next slide. So with that intro, I want to take just a couple minutes here to set the stage, highlighting some of the unique challenges that we all face in oncology development. I think this context is important for understanding the why and the how as to the solutions and strategies that have evolved over time, or in other words, what is difficult in this space and how are we addressing it? So one of the biggest challenges to interpreting oncology data is a lack of healthy volunteer phase, a lack of placebo control in studies. Unique development strategies have evolved to address this. So understanding causality for adverse events, uh, teasing out the role for concurrent treatments or historic treatments, the contribution of any pre-existing comorbid conditions that existed before entering the study. These are all significant challenges that are faced. Additionally, these studies can last many years, often with open label extensions for patients that are responding well to therapy, and that introduces some particularly unique operational and budgetary considerations as well. And the regulatory approval strategies are also quite different here. So pivotal submission data can come from phase 2, for example. Accelerated approval strategies exist that require confirmatory studies to be performed post-approval. So fairly unique in terms of the strategies. The patient populations are also quite unique. Generally, the exclusion criteria for studies in other therapeutic areas uh, allow for enrolling a pretty clean population free of major comorbidities. But oncology is different, and very sick patients with multiple rounds of prior treatments or all the complications from those past treatments, they do enter these studies, and therefore the adverse event rates are higher in these studies versus other therapy areas. But additionally, for these sicker populations, we need to be even more cognizant of patient burden or participant burden. So for any of the technologies we deploy for endpoint collection and studies, this needs to be kept in mind as well. There's a balance to be struck. I think that's obvious in terms of increasing the scientific value of early phase development, adding endpoints that are meaningful, but also considerations for patient centricity, for recruitment, retention feasibility. And lastly, the unique nature of the medications that we study in oncology. So cytotoxic or small molecule treatments, very much still relevant in this space, but there are other more novel mechanisms, including biologics, including immunomodulators, including very targeted therapies to rare or specific genetic mutations, radioligand therapies, hormonal modulators. These are all the different classes or mechanisms of drug development in this space, which each have their own unique challenges. And the last thing I'll mention on this slide, sample size. So relative to other therapy areas, these can be quite small, especially when considering rare tumor subtypes, for example, in a clinical trial population. And so the precision of measurement of any of your endpoints becomes much more relevant when you have small sample sizes. So leveraging the right technologies and solutions that improve the precision and accuracy is really a key theme that you'll hear about from our three speakers today as we talk through this. You can go to the next slide. I'm going to hand over here to Dr. Banzel, who's going to lead us through our, or I should say, who leads our oncology medical imaging group, but is going to take us through the major considerations for imaging in this paradigm and what we can do in an innovative basis to extract even more value. So Anu, I'll hand over to you.
Great, thanks. Thanks for joining, everyone. It's my pleasure to be able to talk to you a little bit today about how to use imaging in early phase oncology. And I'm going to start with just the value proposition for centralized imaging in early phase trials, and I'm going to end with that too. And the reason is, here at Clario, we hear all the time from prospective customers and clients and sponsors that centralized imaging in early phase is a nice to have, but it's not necessary. The data is not usually used for regulatory submission. You can get RECIST information from the sites. And so over the next 15 minutes or so, I'm going to try to explain different things that core labs can do, specifically in early phase, that can aid in decision making. If we go to the next slide, we'll just start with some of the conventional limitations of conventional RECIST assessments, and not that this is new ground that we're covering. People know these limitations, but it will create a nice framework for everything we're going to discuss afterwards. So RECIST relies on static thresholds. It's the categorical variable. We all know this, right? To have a PR, you need a 30% reduction in your sum of diameters. There's a huge stable disease gap, right? That's between 30% reduction in tumor size and a 20% increase in tumor size, all of that stable disease. And all of us know that some of those patients are actually progressing and some of them are responding. Sampling error is an issue with traditional RECIST. People have tried to get around this by sometimes using 10 targets, as in RECIST 1.0, rather than 5. But in early phase oncology, as Todd had mentioned, as Dr. Rudo had mentioned, a lot of the patients have been through multiple lines of therapy. And when they're entering a trial for a new IND, they often have a pretty heavy disease burden. So if you have a patient who has 40 or 50 lesions and you're only following 5, how do you know you're following the ones that are going to eventually demonstrate progression or even response? Baseline blindness we'll get to later in the talk, so we'll skip it for now and go to the next slide. Can we advance the slide, please? Thanks. This is just a little bit of data to start us off. We're going to come back to these concepts, but it highlights this issue of the stable disease gap. So this is data pulled from about 250 patients for 20 phase 1 trials across all kinds of indications. 90% of the patients in this study had stable disease or progressive disease at their first post-baseline evaluation. But when you start to use these other analytic techniques like tumor growth kinetics, total tumor burden, you can see that 82% of the patients who had SD and 65% of even the PD patients demonstrated a response. So we'll use that and we'll start talking a little bit about what these other tools are. So let's go to the next slide. And actually we can go one more further. So we'll start by talking about total tumor burden. And I've used some of these words without defining them. So I want to go back and just make sure the definitions are clear. So total tumor burden is really, it's either the sum of longest diameters or nowadays it's usually volumetric assessment of all measurable lesions in the body. So if a patient has 45 lesions, you get data on all 45 lesions. Tumor growth kinetics is a way to use mathematical modeling to understand the change in tumor size over time. And this can be broken down further. We'll talk about it in more detail in a few minutes, but you can actually separate out things like tumor growth versus tumor decay. Now, why haven't we been using these kinds of metrics? I mean, these aren't new things, right? We've all heard of total tumor burden. Most people probably heard of tumor growth kinetics. The issue has been that for a radiologist to segment this many lesions, for example, and outline annotations on every single slice that they see a lesion on, takes a really long time. We've had studies with machine learning and so forth where we've had radiologists do this task, and sometimes it takes more than 4 hours a time point, which in essence becomes prohibitively expensive and just not feasible with the amount of time it takes. So that's been a limitation in the past, but this image on the left here is an AI algorithm that we developed in-house, and this is showing how it can segment different tumor anatomy in most of the body. And these kinds of tools are what is making this sort of analysis much more feasible and realistic at this point. So let's go to the next slide. So how can you use total tumor burden at baseline? High tumor burden at baseline is actually typically associated with shorter progression-free survival based on imaging and inferior overall survival across lots of different kinds of therapies. We can talk about the immune system. So many medications now are deal with an impact on the immune system. Patients with larger lesions and also a larger volume of overall lesion load can correlate with more immunosuppressive microenvironments, which can blunt the efficacy of different immune checkpoint inhibitors, for example, as well as other medications. Let's go to the next slide. So there's more than one way to use total tumor burden. I think we can see how it's better than RECIST in some ways. I mean, it certainly avoids the sample size problem. One of the ways would be to balance treatment arms, in early phase oncology studies, so that you make sure you don't have one arm that has patients with a much higher tumor burden than others. Which, again, if you're just measuring 5 or 10 lesions, you're not necessarily going to see that. It can even be used as inclusion-exclusion criteria if you only want patients with a certain lesion load on study based on the mechanism of action of the drug or something of that nature. You can even identify high-risk subgroups where standard doses of the standard planned dose of medication might be insufficient based on the volume of tumor that exists. Let's go to the next slide now. So let's talk a little bit about tumor growth kinetics. This is the mathematical modeling I talked about. We'll get more into it, but this is enabled by being able to rapidly generate total tumor burden, because this type of analysis works much better with volumetric imaging, and it works better if you are measuring total lesion burden as opposed to a selection of 5 or 8 or 10 lesions. So the solid line shows the behavior of a tumor, and this is something we're familiar with, right? A patient starts a novel medication, the tumor shrinks, and then over time it starts to grow again, and it grows slowly until it reaches 20% threshold for resistant, and we say the patient has progressive disease. But with mathematical modeling, you can separate out different aspects of tumor behavior from the combined curve. And I have 2 of them here. I mean, there are more things that get parsed out, but just for simplicity, and also these are the most intuitive, you can separate the tumor behavior into tumor growth, which you can see is the dashed line that starts at the bottom left corner and goes up to the right. It just is slow, consistent, slightly exponential growth. And then you can see the other line is tumor decay, where it shrinks and then it sort of plateaus in the amount of shrinking. And so when you combine those 2, you get the total tumor behavior curve. Let's go to the next slide. So why is that important? So what you're really interested in is that G curve, is the growth constant, the growth rate. And that's because that can be directly converted into tumor doubling time. Just to make this, sort of more concrete and less abstract, once you figure out what your G value is, that'll convert into something like the tumor doubling time is 200 days, or it's 500 days, or it's 600 days. But I wanted to talk a little bit about some of the attributes of G modeling and tumor growth kinetics. So the first point I want to make is that G is very highly correlated with imaging-based progression-free survival, and additionally with overall survival. The second point is that tumor growth kinetics correlates with overall survival actually better than RECIST-generated ORR. I mean, let's just think about that for a minute. I mean, how many drugs have been approved based on RECIST determination of ORR? And here we have a biomarker that we're not using most of the time that correlates better with overall survival. And then finally, tumor growth kinetics can predict response and overall survival earlier and with fewer patients. And this speaks directly to what Dr. Rudo was saying with the survey. 60% of our audience here today is concerned with how to find efficacy signals earlier on in a trial. And I'm going to try to convince you that this is one potential answer to that. So we can go to the next slide. The next 3 slides are going to show some data to support these points. These all come from a nice paper written by our friends at Daiichi in Columbia, published a couple of years ago. So I had said that this G constant is very highly correlated with survival. So here we have 2 different study treatment arms, and the patients are separated by quartile of G value. And what you can see pretty clearly is that the lowest quartile has much more inferior survival, and in one of the treatment arms, even the second lowest quartile had much reduced survival. Let's go to the next slide. I made a statement that tumor growth kinetics correlates better with overall survival than ORR does. So this is, again, data from the same trial. On the left, we have a RECIST-based response. Oh, I should say that the plus signs are patients who did not survive, and the plus signs are patients who did survive, and the filled in circles are patients who did not survive. So on the left, we have tumor response across the two treatment arms with a RECIST assessment. And you can see that arm one had a better survival compared to the other arm. And you can see the concordance with overall survival was 0.72. So that's actually not bad. But if you look on the right and you look at what the correlation is between the G value for that same patient population and overall survival, the concordance is 0.82, which is substantially better than RECIST. So that's the second point. Let's go to the next slide. We can talk about the third, which is probably of a lot of interest to people. So how does this work to predict outcomes faster in fewer participants? So let's keep in mind this was a large phase 3 breast cancer trial where this data came from. There are actually two trials, but this is just data from one trial. So powered adequately to show a response with respect to a RECIST assessment, right? So more than 250 patients in each arm. Patients who've been on trial for at least 14 weeks, when they looked at the first 40 patients, we're looking at these two horizontal black lines. They're not that different between arm one and arm two, right? The G value is pretty similar, which again, that translates directly into tumor doubling time. But if you look at the first 60 patients, you suddenly see a pretty big difference between the two treatment arms. And with obviously the treatment on the right is causing better, slower growth, which again translates into better survival. But more interesting though is when you look at the next 80 patients, that difference is maintained, but it's not changed much, right? So in essence, what we're saying is you needed at least 250 patients in each arm to get a adequately powered RECIST assessment, but you might have had your answer once 60 patients have been on trial for 14 weeks. So this is, it's pretty powerful data. Let's go to the next slide. I want to touch on, I'm running out of time, but I want to touch on a couple of other ways where you can use tumor growth kinetics in early phase oncology trials. One of them is dose finding, and this fits in pretty neatly with what the FDA has been saying for the last few years with their Project Optimus mandate, where they really want to move away from max tolerated dose and have a more quantitative assessment of target engagement and rationale for what your recommended phase 2 doses. If you use this type of analysis, we already saw you don't need as many patients, so you could do it in your 3x3 dose escalation, and you can look at the tumor growth rate at different doses. And what you might see is that the max tolerated dose is not the dose that's most efficacious with respect to, again, tumor doubling time. Referential versus experimental, this is what I skipped on the first or second slide, but we'll try to come back to it here. RECIST starts at baseline, right? It starts when a patient enrolls, and then you look for improvement or decline relative to that baseline. But in these early phase oncology trials, most of the patients have had multiple treatments before. So if you can get even 2 scans when the patient was on treatment of another type before they enroll, you can actually do this growth rate analysis on the pretrial scans and compare that to the post-trial scans. So you don't, in that case, need to wait for a 30% change to show a PR. You can show a 10% or 20% improvement compared to the prior therapy. And then finally, event prediction. So you can actually use this analysis to predict future progression events or future response events. And I'll show you a little bit of in-house data in that regard. Let's go to the next slide. This is synthetic data. It's just an example of what I was saying with gemcitabine, where the center column is where you have the best, longest tumor doubling time versus the highest dose that's tolerated. Let's go to the next one. This is some in-house data we were talking about, and we didn't really discuss it, but there's all kinds of different mathematical models that try to, explain tumor behavior. There are models that people use more frequently that really seem to be able to fit at least 90% of patients to a curve, but there are lots of validated models, and so we kind of approached this as a thought experiment and a proof of concept where we were really more interested in the modeling and which models made the most sense to use. So we took two of the most common models and trained a neural network to pick and use whichever model was more applicable to that specific patient within the population. And actually, we sort of did prove our point because the neural network worked better than either model worked alone, and it fit more patients. But leaving that aside, in terms of event prediction, I mean, this was a retrospective analysis of over 400 trial participants with non-small cell lung cancer. All of them had a 2D ROIs placed for resist reads in the past. We have an AI algorithm that can convert those to 3D, and then we use the first 4 on-study time points to try to predict progression or response at the 5th time point. Again, just sort of as a proof of concept. And what you see is that we were able to predict PD versus non-PD with over 70% accuracy, and we were able to predict response versus non-responders with over 80% accuracy. So as you think about it, I mean, this is a small patient population. We're just starting to train this neural network. Our plan is to incorporate all of the 7 to 8 most common mathematical models and train on a much larger data set. But to be able to predict response and predict PFS events is pretty powerful stuff. So the last slide, the next slide is my last slide. I just want to get sort of summarize. I think the message I've been trying to send is that because we can now use artificial intelligence algorithms to generate total volumetric tumor segmentation for us, that's very powerful because it enables lots of other kinds of analysis that were previously really too cumbersome and impractical to do on a larger scale. So again, just the last point here, it's early phase oncology imaging. Everybody is collecting all the information they need to derive this in any study because it's just the standard of care imaging. So if you're not looking at some of these data points, you're leaving a lot of valuable information on the table. With that, I'm going to hand over to my colleague, Dr. Dumais, and she's going to talk about ECOA.
Great. Thanks so much, Anu. Hi, everyone. Very happy to be here. Thanks for joining. So you've just heard of the value and power of these imaging techniques. I'm going to switch gears and have you think about the value of incorporating subjective patient experiences into early phase oncology trials and doing that through the use of patient-reported outcomes, so PROs. Because what we're hearing more and more from regulatory agencies and patient advocates as well is that patients want more of a voice in reporting their symptoms and experiences, especially in early phase, and that regulators are wanting this information for benefit risk evaluation too. So next slide, please. So how do you develop a meaningful PRO strategy? The first step is what are regulators recommending, right? So let's look to see what regulators are recommending when it comes to which PROs to include in oncology trials. This might be familiar to a lot of you on the call. This is the FDA Core PROs in Cancer Clinical Trials Guidance, which was first released in draft in 2021. It has now been finalized since 2024. It emphasizes the added value of incorporating PROs into oncology trials and really the importance of this systematic assessment of these core PROs. And they recommend these core set of 5 PROs for including in oncology trials. So disease-related symptoms, physical function, role function, symptomatic adverse events, and overall side effect summary measure. So the physical function and role function are commonly measured by assessments such as the EORTC QLQ-C30 or the functional assessment of cancer therapy, the FACT-G. The disease-related symptoms are commonly assessed using subscales or cancer-specific modules of those assessments. Symptomatic adverse events are commonly assessed using the PRO CTCAE, which we'll talk a little bit about later, and the overall side effect impact measure is commonly assessed given just a one item assessment in the FACET scales or the EORTC item library has a side effect burden measure. So, and another main component of this guidance is that it also recommends more frequent assessments of PROS. So more than that kind of traditional, less frequent at-site assessment, it really recommends as much as weekly assessments for some of these core PROS at early treatment cycles. And while this guidance is unspecific in terms of what phase of oncology trials that you're running, if we go to the next slide, this one does specify the importance of PROS in early phase. So you can see that the FDA does support the use of PROS in early phase. This is their guidance on optimizing the dosage of oncology drugs, also finalized in 2024. And while this is not specific to PROs, it's not a PRO guidance, it lists PROs as a recommended data that should be used as part of optimizing dosage. So it states that PROS can provide a systematic and quantitative assessment of expected symptomatic side effects and their impact on function, and should be considered to enhance the assessment of tolerability. And I'll emphasize the note on impact on function, right? Because this is especially something that is not very well understood or assessed by clinician reports alone. It is really patient-reported outcomes that really best describe the symptom impact on functioning and daily functioning. And I'll also emphasize that while this additional PRO data is important, it's complementary, right? So it doesn't replace clinician reports, but it's complementary to clinician-reported adverse events and tolerability data. Next slide, please. So when in drug development programs for oncology should you be incorporating PROS? The answer is as early as possible, and this is what the FDA has stated. So if you do not attend regularly already, I highly recommend you bookmark FDA's annual COA and Cancer Clinical Trials workshops. These are virtual, public, free to attend, and always have a fantastic lineup of academics and patient advocates, FDA representatives. And it's really where they're sharing their latest thinking around PROS in oncology. And I've attended every year for the past several years, and I can say incorporating PROS in early phase has been one of the themes and points of emphasis in the past couple of years. This is for a few reasons. The first, it's a way to ensure patient centricity, right? In that it's early phase is the first opportunity to formally include the patient voice. PROS are the gold standard method to measure subjective experiences. So why wouldn't they be included in oncology, which is a highly symptomatic, life-altering state disease? Secondly, it is your opportunity to also optimize later phase PROS strategies. So early detection of symptomatic side effects or early detection of improvements in symptoms or quality of life will allow you to better focus and optimize your PROS strategy for phase 2 and 3, and could potentially reveal opportunities for drug differentiation in, as we know, a highly competitive market. Thirdly, it will allow you to be submission ready. So we know that with accelerated approvals, it's not uncommon for oncology treatments to be approved using non-phase 3 data. So incorporating PROS early in your drug development program will also allow you to be in line with regulatory expectations. Finally, including PROS will allow you to better evaluate tolerability, which is we know one of the major objectives of phase 1 trials, and a better evaluation of tolerability will help reduce and can help reduce the need for example, dose reduction or reduce later patient dropout due to intolerable toxicities in later phases. Next slide, please. So to dive into this concept of tolerability a little bit more, I love to show this article. And while it's not a new publication, it was released in 2010, I think it encapsulates very well the pitfalls of relying solely on clinician-reported data for assessment of side effects and tolerability. The current traditional practice for collecting AEs and toxicities assumes that clinicians have an accurate picture of the patient subjective experiences, right? But we know that often this isn't the case. And there's really no standardized method for how clinicians are assessing this. So this study directly compared clinician-reported AEs with patient-reported symptoms, and what they found was that clinicians tended to systematically downgrade the severity of patient symptoms, detect symptoms later, miss symptoms altogether. So when we think about, an early phase oncology study where the primary objective is assessing safety and tolerability, define that recommended phase 2 dose, it's really important to be including the patient voice in this, as they have the most accurate information on their symptoms and able to report them more accurately. Hot off the press this month, also in the Lancet, is a paper that looked at inter-rater reliability of clinician AE assessments with or without clinician access to review of PRO data. And what they actually found was that investigators, when they had access to PRO data while grading AEs in oncology trials, the consistency of those AE ratings improved significantly. So raters were better able to characterize adverse events when they had access to PRO data. So that was hot off the press. I suggest you get to that paper too. Next slide, please. So this is kind of a summary of, again, why use PROs to evaluate tolerability, and again, why it's so important to include PROs in this early phase. Hopefully I've convinced you already that you can, get more precise, sensitive, accurate data with PROs. Not only can clinicians underreport symptoms, but miss symptoms altogether. You also get more granular data with PRO data, such as the PRO CTCAE, because you're evaluating not only the presence, but also the severity, the frequency, the interference on daily activities. And again, this is really data best known and reported by the patient. Next slide, please. So this is a little bit of a, don't just take my word for it, right? There have been a few pivotal publications in the past couple of years that show consensus thinking and recommendations on the topic of incorporating PROs in dose finding early phase oncology trials. And the general consensus is that there is agreement among stakeholders, and these stakeholders included academic researchers, industry reps, patient advocates, PRO methodologists, even regulators, that PROs should be reviewed when making dose escalation decisions and determining the maximum tolerated dose or recommended phase 2 dose. And while I won't pretend that there aren't some challenges to be considered, operational considerations, given this idea of early phase PROs is still relatively in its infancy, there is clear, I think, agreement upon the benefit of using PROs in these phase 1 trials, including that they can aid clinicians in AE reporting, detect early AEs, minimize dose modifications later on, et cetera. So the benefits are really clear, and we're still working through those operational considerations. Next slide, please. And this is just one more publication, which has just been released last month, that affirms, again, the consensus recommendations of key stakeholders on the importance of incorporating PROs in early phase, but also gives more specific recommendations on why and how to do so. So in my first slide, you can recall that there were 5 core PRO categories. This paper is the first to recommend a subset of these for use in specifically early phase oncology, recommending that sponsors focus on side effect impact, symptomatic adverse events, and overall health-related quality of life. So they go further into the recommendation of which PROs to use and in what settings, et cetera. So another great resource for you. Go to my next slide. As I begin to kind of wrap up my section, my remaining couple of points are geared towards operationally, right? How do we incorporate PROs in early phase? So at face value, PROs administered on paper might seem the easiest solution. There are a lot of considerations when doing this, including the negative impact on data quality, backfilling by patients, higher risk of missing data, incomplete, added variability, et cetera. So we know very well the added value in terms of data quality when it comes to eCOA. And this is just to say, even the FDA and EMA oncology specific guidances do point out the benefits of electronic data capture for oncology trials, stating its potential to lessen patient burden, adding convenience to patients, et cetera. Next slide, please. Your next thought might be, well, eCOA is great in theory, but it's really not feasible for phase 1. This is a great publication that came out also just last year that shows independent evidence supporting the use of eCOA in early phase oncology. So it included weekly remote PROs and found really great compliance at over 85% plus compliance. Found that it captured 3 times more symptoms than the clinician reported CTCAE, and again, identified symptoms that were often missed by clinicians. So again, just a great example of the benefit of not only PROs, but also the feasibility of doing ePRO, so electronic PROs in early phase oncology too. If you go to my next slide, this is my last slide just to wrap up. How do we then implement eCOA for using these ePROs in early phase? This just shows the many options that are available. You'll commonly see provision devices that patients can take home. They can use their own device, BYOD. You may also commonly see tablets or web being used at the site. And the key here is that we really just want to make sure that technology is flexible and easy to use, fits in easy with their lifestyles and their expectations for how technology should look and function. The exact, device strategy is based on a number of different aspects. Training participants on how to use devices and also the importance of pros will also be really important for any ECOA or ePro strategy in early phase. So that's all I have time for today for my section. I hope this was helpful for you as you consider how and when to incorporate PROs and ECOA in your early phase oncology study. And I will now hand it off to Dr. Kleinman.
Kelly, thanks everyone for joining us. I'm going to talk a little bit about cardiac safety and try and explain the advantages of assessing cardiac safety in the first-in-human studies. Next slide. So regulatory requirements are that we assess cardiac safety of all new drugs, and the reasons are pretty simple. Any drug, no matter what the route of administration, if it is going to get to a distant part of the body, it has to do that through the blood, and all the blood goes through the heart. So cardiac cells are exposed to any drug that has systemic bioavailability, and therefore cardiac toxicity, even though off target, is theoretically possible, and you therefore have to exclude it in your clinical trials. Next slide. And oncology drugs are, as everyone knows, different in a lot of ways than non-oncologic agents. A lot of them have cardiac toxicities. And a regulator considering approval of a new oncologic has to weigh the risks and benefits. And of course, in oncology, the balance is shifted towards benefit. Side effects are expected, and most can be managed. Obviously, there's not really much benefit to a patient if their tumor shrinks, but they develop torsade and sudden death. But again, looking at this in risk-benefit analysis, if we exclude a treatment that has a 1 in 10,000 risk of causing sudden death for a tumor that has no other treatment, who are we treating? We're really not doing what's best for the patients. Next slide. So this is a list of the 28 FDA oncology new drug approvals in 2024 and 2025. And there are a variety of different molecule types and disease states. Next slide. And in the right column, I've listed the drugs that have cardiovascular toxicities, and 10 of the 48 did. Most of them were QTc prolongation, but there were also some that produced hypertension, bradycardia, and left ventricular dysfunction and heart failure. Next slide. When I talk to people in pharma about cardiac safety, they often look at me and say, are we just talking about QT? Well, no, we're not talking just about QT, but QT is certainly a very large concern in any new development program. Next slide. And this is exactly why. This is a patient who developed torsade de pointes and deteriorated into ventricular fibrillation. This was caught on a Holter of a patient who died. And this was related to drug-induced QT prolongation, which is the setup for torsade, and torsade is often fatal. Next slide. Now, in oncology, the poster child is arsenic trioxide. And yes, that's the same arsenic in the movie Arsenic and Old Lace. And obviously, it is a poison. That's why people use it to murder people. The efficacy of arsenic trioxide was demonstrated for promyelocytic leukemia, however, and the cardiac toxicities weren't initially appreciated. But retrospectively, we went back and evaluated the ECGs that have been collected in the clinical trials and found that arsenic trioxide is one of the most potent QT prolonging drugs imaginable. The mean QTc increase was 47 milliseconds, Nearly half of the patients had a QTc above 500, which in most clinical trials is a criteria for stopping treatment. Nevertheless, it's approved for use because, again, risk-benefit. You can monitor the QT interval, adjust the dose if necessary, and ensure patient safety. Next slide. So the current oncology division guidance is that you have to characterize the ECG effects of new oncologic agents, and it's really to inform prescribers about how to use the drug safely. Cardiovascular toxicities in oncology are usually a labeling issue, even for drugs with a very potent effect. And again, the purpose of the label is to let prescribers, once the drug is approved, understand how to use it, how to monitor patients, how to do it safely. Obviously, if we learn this early in drug development, we can use the same guidance in our later phase trials. Next slide. So when I talk to clients, they often want to discuss what's the best timing to assess cardiac toxicities, and particularly QT prolongation. Should we do it in our first in human studies, or should we wait to see if we get to phase 3? And obviously, we all know that most drugs that enter first in human trials do not make it to NDA. And there's a reluctance to overload the first in human studies with assessments that really aren't going to be needed unless the drug makes it to phase 3 and NDA. If a drug is abandoned and put on the shelf, it's really not terribly relevant if it prolongs QT or not. So there's sometimes a thought maybe we can wait till phase 3 to assess QT. The problem with that is the tool that we use for QT assessment in most oncology trials is concentration QTc modeling, not a by time point analysis. And that requires adequate power and sufficient PK. Now, first in human studies in oncology, which are usually phase 1/2, have the widest range of doses and exposures that will ever be encountered in any of these trials. And often they may have a super therapeutic dose and exposure that will not be evaluated subsequently. But most important, the first in human oncology trials are usually the only trials that have intense PK. And we need that for the assessments. In contrast, if you try and do a late phase study and have a QT sub-study that requires intense PK, it's extremely difficult to recruit. And I've seen many of these fail to meet their recruitment goals. Next slide. So in general, what we recommend is to try and do this in phase 1/2 studies, because that's where you're already collecting the intense PK, and all that you need to do is bolt on additional ECGs. And the ECGs need to be time matched to the PK. And obviously, the patient is still at the clinic, at the site, in order to have the PK drawn. So we're not requiring them to have extra visits or stay longer to get their ECG assessments. Again, since the cohorts are going to be small, we'll use concentration QT assessment. And we usually like to make sure we get assessments both with the first day of dosing, as well as when the drug and or any metabolites are at steady state, both to look for delayed effects from metabolites or delayed off-target effects that aren't related to hERG binding. And very important to emphasize, the purpose of monitoring QT for an oncologic program is not that of a thorough QT study, which is to detect a small QTc effect. The purpose of the monitoring is to exclude a large QT effect. Next slide. I'm often asked why centralize ECGs in oncology trials? Can't the oncologists use them and handle them themselves? Next slide. Well, to look at that, we did a study where I looked at 270,000 consecutive ECGs from clinical trials in oncology, for which we had both the automated measurements that were provided by the ECG printout and that most sites rely on, as well as the centralized measurements. Huge number of samples, huge number of patients, almost 300 different studies. Next slide. And what we found is that you really should not be entirely comfortable with the ECG machine reported measurements. When the ECG machine reported that the QTCF was greater than 450, that was a false positive in 30% of the cases. And when the ECG machine reported a QTCF above 500, 77% were false positives. The difference between the correct measurements and the automated measurements wasn't a millisecond or two. The mean difference was 80 milliseconds. That's reporting a QTC of 500 when the QTC is really 420. And false positives are bad for programs and bad for patients. They may exclude a patient who really should be eligible for a trial, and they may interrupt therapy inappropriately during a trial, which is not good for anyone. Next slide. And needless to say, even worse, there are false negatives. When we measured a QTCF of above 500, which in many studies is reason to withhold a dose, the ECG machines missed that in almost 56% of cases. And again, they missed it by 60 milliseconds. It wasn't close. So we believe that it really is important to get accurate data, both for patient safety and to ensure that you get the right answers for your program and don't get a false or positive negative assessment. Next slide. Obviously, QT prolongation is not the only toxicity that we encounter. Many oncologic agents produce heart failure or pericarditis, heart block, atrial fibrillation, changes in blood pressure, and those we don't identify on 12-lead ECG. We look at other modalities. Next slide. As an example, VEGF inhibitors as a class, they may produce hypertension, LV dysfunction, thrombosis, QT prolongation. Next slide. TKIs, same story, same types of side effects. Next slide. I mentioned blood pressure. There are a number of different modalities in what can use to assess the effects of an oncologic on blood pressure: clinic, home, or the most precise ambulatory blood pressure monitoring. And again, you'll tailor that based on the non-clinical and clinical data that you obtain. Next slide. So the best practices are, first, you need to do ECG assessments in all phases, often because these are older patients with underlying cardiac disease. And irrespective of their treatment, they may have cardiac events that you need to follow. In contrast, we would only recommend assessing these if there's some reason to expect it, a class effect or a non-clinical signal. Serial troponins, blood pressure assessments, serial assessments of left ventricular function. And that is my last slide. And we'll turn it over to Dr. Rudo.
Okay, perfect. Well, thanks, Dr. Kleinman. Thanks, of course, to Dr. Benzel, Dr. Dumais as well for your insights today. That does bring us to the conclusion of our prepared content today. I think actually, rather than take the time to recap all the key points from what you've heard today, which is what is on this slide, Samia, why don't you go to the next slide? I think it'd be best if we just jump right into the Q&A with the remaining time here. So I'll invite all the speakers to come back on screen. Certainly appreciate everyone's time and attention. Glad to see so many questions that have come in as well. So I'm going to just jump right there. So first one here, I think best to Dr. Benzel. Let's see, can you clarify how radiomics fits into the tumor growth kinetics discussion, and what type of analysis or predictions can this be used for in early development? So radiomics.
That's a great question. Radiomics actually it fits very neatly. I would say it's adjacent to tumor growth kinetics. So it's something that is facilitated by the AI-based total tumor burden segmentation that I talked about, and you can use those that data, those ROIs. You can use that and apply it to radiomics analysis. And radiomics, just briefly, I mean, it generates different types of features that are not really visible to the eye. Some of these have been correlated with tumor aggressiveness, the risk of tumor metastasis, and other attributes like that. It's still a little bit early, but there are lots of papers and there's a lot of research being done on how to best use radiomics. But I would think of it as something that's adjacent to tumor growth kinetics.
Great. Yep. Super helpful. Thanks. Next one here for Dr. Dumais. It's a good one. So when assigning patient-reported outcomes to patients for completion outside of the clinic, how can you encourage or ensure compliance so there isn't a lot of missing data? How can you assess the burden to patients when adding PROs in early phase like this? So compliance and burden, I think, are the two themes here.
Yeah, and those are two common themed questions I feel we get often, because we know, how sick patients can be, and especially in these early phase trials, we do want to ensure we're keeping burden low. So I'll start by saying, patients again want to report how they're feeling, so they want that voice in trial. So it's not the pros themselves that are burdensome. Where it becomes burdensome, I think, is when we throw too many PROs at them, or too many repetitive questions at them, or give them pros without explaining to them why they're important. So ways to minimize patient burden will be, as I mentioned briefly before, training patients so they know not only having the technical support on how to complete them, but also why they're completing them. This can really help with engagement. Also needing to ensure you have the right pro strategy, right? And that you're thoughtfully including the pros that are only required and fit for purpose and necessary for your study, to keep the number down, and also allowing flexibility and how and where they complete pros. You mentioned at-home completion, and that's one way to minimize burden, allowing them to complete pros in the comfort of their own home, perhaps on a BYOD, bring your own device strategy. And there are many strategies for making sure compliance is still high when they're completing pros at home, like alarms and notifications, and again, just keeping the ease of use as easy as possible. The design of the eCOA and the app itself. So there's many ways to kind of reduce burden. So we just need to make sure that we're having thoughtful kind of pro strategies to reduce that burden and keep compliance high.
Okay, perfect. Yeah, some important considerations there, definitely. Dr. Kleinman, we're going to squeeze in one more here for you. This is about different types of drugs. So are dedicated QT assessments needed for large molecules such as monoclonal antibodies or antibody drug conjugates, or is that mostly applicable for traditional small molecule programs only?
Thanks. That's something that comes up quite a bit. And as a general rule of thumb, the level of QT assessment and cardiac safety assessment that you need to do is inversely proportional to the molecule's size. So at one extreme, monoclonal antibodies are so large that there's no concern about them directly prolonging QT. Still have to do some ECG assessments to make sure there aren't indirect effects, but the level of concern is very low. For antibody drug conjugates, the level of assessment would depend on whether the linker or the payload are detectable in the systemic circulation. If they are, you may be asked to do a full QT evaluation of those. And peptides and oligonucleotides are something there is no guidance on yet. Hopefully the ICH will release a guidance about that in the next few years, but for the moment, there is a requirement for ECG assessments that are not very much different than for a typical small molecule.
Okay, great. Really helpful. Thanks. Salmi, I think I'll turn over to you now to close us out as we are at the top of the hour. But big thanks to our speakers here, certainly, for joining me today. And as I said, turn back over to you.
Thank you so much, Dr. Rodo, and thank you all for those very thoughtful responses. That brings us to the end of the Q&A portion of today's webinar. Before we close, I just have a few final notes to share. For those who are interested in hearing more from Buclario, you can register for their next webinar, Novel Digital Endpoint Solutions for Early and Mid-Phase Development, and that's scheduled for Tuesday, March 10th, and I've sent the link in the chat box as well. And if we couldn't attend to your questions, the team at Baclario may follow up with you. Or if you have further questions, please feel free to direct them to the email address on your screen. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen, and your participation is greatly appreciated as it will help us to improve our webinars. Additionally, I have also shared a link to view the recording of today's webinar in the chat box, and you can also share with your colleagues so they may register for the recording here as well. For those watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and gain access to features such as viewing the recording and more. Now, please join us in thanking our speakers for their time here today. We hope you found this webinar informative. Have a great day, everyone, and thank you for coming.
Thanks.
Thank you.